Hello, BISC130. This is the beginning of recorded lecture 2.5, and a, a bit of a break after the last couple of chapters. Very heavy, complicated stuff in cellular respiration and photosynthesis. This is going to be more straightforward in comparison. We're going to zoom out quite a bit now. Instead of talking about individual cells and how they process energy in different ways, we're going to talk about how multiple cells will talk with one another. Cell communication. So there is one very important key term to define here at the top, because it's going to get used throughout this chapter, and that is the term ligand. It's also pronounced ligand. That's an envelope sort of thing. Either one is fine. I think I usually say ligand, but now that I'm pointing it out, I'm in my head about it, and I'll probably alternate between ligand and ligand. It is what it is. So what is a ligand? Well, to read from the key terms, a ligand is a molecule, again, very generic, just calling it a molecule produced by a signaling cell that binds with a specific receptor, delivering a signal in the process. In other words, this is a very generic term for any sort of molecule that sends a message. Ligands are released from signaling cells, and they bind to receptor proteins on target cells. And here's a good visual of a couple of different ways that this can happen. Here's our generic ligand. Here is a receptor. Here's just that happening again. Here's a ligand. Here's a receptor. And yeah, it sends this message. Stuff happens, and the cell behaves differently as a result of that signal. But again, here's a ligand out here, a signaling molecule. Now, there are 4 types of signaling mechanisms that we could break down based on where the ligand goes and how far the ligand goes. So I'm going to number these as usual. You know, my test will not say, what's number 3? The numbering is just so we can count to 4. So one of these is called autocrine signaling. Let's take a look at this before I write anything down. Cell talks to itself. So, I mean, just like you could hear the sound of your own voice, cells can send out ligands and, receive that message themselves as well. So autocrine signaling is very simply when the ligand binds to a receptor on the cell that sent it out, on the signaling cell. This happens often in virus-infected cells. Cells that are infected with viruses need to kill themselves, very grim, but very important to prevent the spread of that virus, and they'll send this sort of message out to itself in order to make that happen. Mechanism number 2, direct signaling. So once again, I got a figure for this. As the name implies, direct signaling is just 2 cells that are directly talking to one another. Usually this ligand in our other forms of communication has to travel some sort of distance, has to float around to the other cell. In direct signaling, the 2 cells are directly touching one another. The ligand is sent from one cell directly to the other. Here's my summary. Ligand travels from the cytoplasm to cytoplasm, from signaling cell to the target cell. This sort of signaling is done in the heart, where you have cells that really need to talk to one another very, very, very quickly, and they'll communicate with one another through this type of direct signaling. Number 3, we've got paracrine. So this is where the ligand goes from one cell to a different cell, so it's not autocrine, and the ligand does have to travel a distance. They're not touching one another, but it is a short distance. So signaling cell sends out a ligand, it travels a short distance where it binds to its target cell or cells. To summarize, in paracrine, ligand binds to receptors on nearby cells, and I've underlined the word nearby. Again, it's important to note that in this type of signaling, the message does not travel far. The ligand itself, whatever it happens to be, just doesn't have a good shelf life. It is rapidly degraded. That makes sure that the signal is local. So this ligand is not going to send messages throughout the entire body because it just doesn't last that long. It's only going to send its message to nearby cells. A good example of paracrine signaling is in the immune system at a wound site. So if you get a cut or a scrape or something like that, those damaged cells are going to send out ligands saying, hey, we have a breach of security here, the skin has been damaged, we need to bring in some immune cells to deal with the inevitable pathogens that have gotten underneath the skin. And yeah, when that happens, you don't want these ligands sending the entire body's immune system into a storm. You just want to target the stuff that's nearby, saying, hey, send in the troops. We've got a wound site here. We've got some invaders here. Take care of business. So paracrine, short-lived signal, nearby response. In contrast, we have our fourth type called endocrine. During endocrine signaling, we have a signaling cell, ligands, traveling a long distance in order to hit their target cell or cells. So yeah, this says bloodstream in many animals. Yep, the way you get from one cell to another cell, if they are far apart from one another, is to use the bloodstream. So endocrine, ligand binds receptor on distant cells, and as opposed to being short-lived or rapidly degraded, here we have a stable ligand that ensures that it can make that trip, it can travel across the body. A good example of this is lots of different hormones, and we've talked about hormones in previous chapters. Many hormones are produced by the thyroid or the adenoid glands, or all different parts of the body, and the cells that they need to actually affect are somewhere distant from that site of origin. So yeah, endocrine signaling, there are many hormones that use this type of signaling. And I broke down the four, but here's a nice slide that shows all four of them together: autocrine, direct signaling, paracrine, endocrine. Now, these were four types based on where the ligand goes and how far it goes. In addition to this, we could break down cell signaling into two types based on where the receptor is. So again, just based on different criteria. This is how the ligand travels. These two types now are where the receptor is. The first one I want to bring up is called an intracellular receptor. This figure should look familiar. I cut out the other type. We're going to focus just on this one. An intracellular receptor is a protein receptor that is inside of the cell, in the cytoplasm. Now, in order for the ligand to bind to the receptor, again, that's the point of this whole thing, ligand needs to bind to receptor. If a ligand needs to bind to a receptor and the receptor is in the cytoplasm, that means the ligand needs to cross the plasma membrane. That is not easy to do. Again, we talked about plasma membrane in several earlier chapters. This is a pretty effective barrier. This is a pretty effective wall. It prevents most stuff from coming in or out. But in intracellular signaling, it's got to cross this barrier. It is possible to do this. There are certain steroid hormones. Hey, we talked about steroids in an earlier chapter, this sort of four-fused ring structure here. This type of structure that is largely hydrophobic but has a little bit of hydrophilic to it, this has just the right kind of chemical properties to be able to get into the bilayer and then out of the bilayer again into the cell. So this is the type of signaling here, and these ligands have to have those kinds of properties to make this happen. In summary, intracellular receptors are where the receptor protein is in the cytoplasm of the cell, and ligands for these receptors need to be small and hydrophobic in order to diffuse across the membrane. An example of these ligands is steroid hormones. So in contrast, so this was type number one, the intracellular receptor. Other type is what we call a cell surface receptor, which, as the name implies, the receptor is on the surface of the cell. So here we go, the other half of that figure. This is not floating around in the cytoplasm. In this type of cell signaling, the receptor is embedded within that lipid bilayer, within that membrane. It is an integral protein, or integral membrane protein, to use a term from an earlier chapter. And when the receptor is in the membrane facing the outside world, the ligand, whatever it is, doesn't have to cross the membrane. It just needs to bind to the portion of this receptor that's facing the outside world. So receptor in cell surface receptors, the receptor protein is on the surface of the cell, anchored within the plasma membrane, and in order to do its job, it actually needs 3 distinct domains. We didn't talk about domains when we talked about protein structure. These are just different structural regions of a protein. So you need 3 different parts in order for this job to happen. And here's a different figure that sort of breaks these parts down. These cell surface receptors need to have an extracellular domain. That's the part of the protein that faces the outside world. That's the part of the protein that ligands are going to bind to. So, 3 distinct domains. Extracellular domain, exposed to the outside, binds ligand. Simple enough. The other domain that any cell surface receptor needs to have is something called a transmembrane domain. Trans means across or crossing. Membrane refers to this membrane. So going across the membrane, this is the domain of the protein that is hydrophobic and is going to anchor this entire protein where it's supposed to be in this lipid bilayer. So transmembrane domain, it spans the membrane. And our third part is the cytoplasmic domain. This is the part of the protein that faces the cytoplasm. So we're just talking about ligand binding to receptor at this point, but eventually, soon, we're going to talk about these receptors actually doing stuff, continuing that message on through the cell. And in order to do that, part of this protein does need to be interacting with other stuff in the cytoplasm. So we need a cytoplasmic domain, a domain that's exposed to the cytoplasm. And again, we've got all 3 of them shown here: extracellular, transmembrane, cytoplasmic. Now, when we were talking about intracellular receptors, I told you that these ligands needed to have properties like this in order to do their job. However, in cell surface receptors, these ligands can have all sorts of shapes and sizes. They don't need to have special properties to cross this tricky membrane. You could have massive proteins that are these ligands, like insulin here. You could have lots of other chemical compounds. Again, they don't need the specific chemical properties of crossing a membrane. Cell surface receptor ligands come in all different shapes and sizes. They just need to be able to bind to the extracellular domain of this receptor. So ligands for these receptors, cell surface receptors, are water soluble. They're floating around in the water, and these include small molecules or proteins. Again, very generic because, they come in all different shapes and sizes. They don't need to have special properties. So what happens next? Our ligand has bound to our receptor, one way or the other. What's the next step? Well, the next step is something called signal transduction. So the binding of the ligand to the receptor leads to a change in the shape of the receptor. We're going to see this throughout this quarter. Anytime you mess with a protein, anytime you add something to a protein, anytime you bind something to a protein, anytime you just add a little chemical group or add a partner or whatever, anytime you mess with a protein in any way, you're going to change its shape just a little bit. For proteins, shape is everything. If they have a different shape, they're going to behave in a different way. So binding of ligand leads to change in the shape of the receptor, and it's going to behave differently. What do I mean by behave differently? Well, here's one example of this. So we're looking at an intracellular receptor here. There's our signaling molecule, our ligand, crossing the lipid bilayer, binding to this intracellular receptor, and without its ligand, this receptor was just floating around doing nothing. But now that it has ligand bound to it, it is going to behave differently. It's going to do something different. In this case, it is going to enter the nucleus of the cell, bind to DNA, and turn certain genes on or certain genes off. It depends on what the message is, but it's going to direct the synthesis of certain proteins from the DNA blueprint or stop making certain proteins from the DNA blueprint. And again, it's only doing this with the ligand bound. It wasn't doing this when the ligand wasn't bound. So this is an example. Not all signal transduction works like this. This is just one way this can happen. So to behave differently could be in an intracellular receptor, it may enter the nucleus and cause genes to be turned on or off. That's definitely going to make the cell behave differently. But that's not the only type of different behavior that can happen. Sometimes the binding of ligand to the receptor leads to the creation or release of what are called second messengers. And again, the name is right on the money here. If the ligand is the first message, the creation or release of just more messenger things, these things are called second messengers. They're going to go off, and depending on exactly which one we're talking about, send further messages within the cell. So sometimes this ligand plus receptor just leads to more messages being sent in the cell. So this may lead to the release or creation of a second messenger, which is defined in the key terms. Second messenger is a small non-protein molecule. Again, these things don't memorize their structures or even their names, but these are small non-protein molecules. Second messenger is a small non-protein molecule that propagates a signal within the cell after activation of a receptor causes its release. So just something that continues sending this message, a second messenger. But that's not the only thing that can happen. Another possibility for how things can behave differently once this ligand has bound to receptor is phosphorylation. So specifically in a cell surface receptor, this binding event may lead to the phosphorylation of a protein or proteins. Now, at this point, we have to do a bit of a sidebar here. So phosphorylate is an important term. It is not in the key terms because I want to write this out, myself. Phosphorylation involves a phosphate group, as maybe you could infer based on the name. Phosphorylation is where we take a phosphate group from ATP, take it off of ATP, it becomes ADP, and we attach that phosphate group to the amino acid of a protein. So phosphorylation is when we attach a phosphate group to a protein. If I want to define that more formally, phosphorylation is the modification of a protein by the addition of a phosphate group or groups, this is showing just one, sometimes it's several, from ATP. Importantly, this is a very widespread type of chemical reaction. There are many phosphorylations that occur in many different proteins throughout the cell. And so the enzymes that do this, because this is not magically happening on its own, there's an enzyme that's going to catalyze this chemical reaction. Any enzyme that performs phosphorylation is called a kinase. That's just the name for this class of enzymes. So phosphorylation is catalyzed by a class of enzymes called kinases. There are many of these in the cell. Now, we have phosphorylation to add a phosphate group. There's another side to this coin. We also have a process that can remove a phosphate group. This process is appropriately called dephosphorylation. You got a protein with a phosphate group attached, you remove that phosphate group, now you have an unphosphorylated protein. So dephosphorylation is the removal of a phosphate group or groups from a protein. Again, there are many enzymes that make this happen, and I wish they were called D-kinases. It would make a lot of sense if they were D-kinases. Unfortunately for us, they are not called D-kinases. The class of enzyme that performs dephosphorylation is a phosphatase. Again, this is a big umbrella category. Many phosphatase enzymes exist within the cell. So dephosphorylation is catalyzed by a class of enzyme called phosphatases. Now, this is all just terminology about phosphorylation and kinases and dephosphorylation and phosphatases. The point of all of this is this statement. The function of a protein can be switched on or off depending on its phosphorylation state. So, just like a light switch in a room, you can shut a protein on or off by adding or removing a phosphate group. This is a lot faster than having to, wait to build new proteins or stop building certain proteins. This is a way to make the cell behave differently, but it takes a while. Again, second messengers can, kind of take a while to send their message to do something else. Phosphorylation and dephosphorylation are great, quick ways for the cell to immediately behave differently. By adding or removing this phosphate group, you control whether this protein behaves a certain way or doesn't behave that way. I wish I could tell you that, one of these was always on and the other one was always off, but it completely depends on the protein. Some proteins, their activity is turned on when they're phosphorylated and they're off when they don't have a phosphate, but some are the exact opposite. Some are off when they don't have a phosphate, and adding that phosphate switches them the other direction. So that's why I'm being sort of generic here when I say the function of a protein can be switched one way or the other depending on its phosphorylation state. So all of this was so that we understand this statement a little bit better. Ligand plus receptor can lead to phosphorylation from the cell surface receptor. It's going to lead to stuff behaving differently. Here is a figure showing this. Ligand binds to receptor. The receptor, first it phosphorylates itself in this particular example, but then it goes off to phosphorylate other proteins, which are going to behave differently now that they have been phosphorylated, and that's going to lead to a cell response. Now, you may have noticed that I've been very, kind of vague so far with many of the descriptions in this chapter. I haven't called out the names of any of these receptors. I haven't, described exactly what happens in the cell. The reason why I have been so vague is to go into the specifics of, a named signaling pathway is going to get very complicated. So let me show you what I mean. Let's take a look at a specific signaling pathway, what this looks like with named ligands and receptors and how they interact with one another in the cell. Let's take a look at an example. Oh yeah, we're not doing this in an introductory biology class, but I am showing this to you, this MAP kinase signaling pathway to show how complicated this gets. There are so many all these are different ligands that can activate different receptors, and you lead to activation of different proteins, which lead to activation of different proteins and different proteins, all these different pathways leading to a cell response, and how they all intersect with one another and overlap with one another. We have several different cell responses here at the bottom, things that can happen at the very end. But yeah, this is why I'm not doing specific examples in this chapter. Almost any specific example you want to get into is going to get out of hand very quickly. So yeah, I'd like figures like this just to help you appreciate how complicated this truly gets. But yeah, that's why I'm being vague. So we talked about binding to receptors, what the different behavior is. Let's skip to the end though. What's the end goal of any of these pathways? So what's the final step? What's the ultimate result of this? So this signal transduction, ligand binds to receptor, you get second messengers or phosphorylation, but what's the end goal here? Well, this can ultimately result in altered cell behavior. And again, I'm being generic here. The cell can, produce proteins that it wasn't producing before. It can move in a way that it wasn't moving before. It can, take things from the environment that it wasn't taking from the environment before. Altered cell behavior. Another ultimate result of all this stuff is cell growth or division. So we'll talk about cell division in a couple of later chapters coming up, but this cell division and cell growth is controlled by pathways like this. Sometimes the ultimate result of a ligand binding to a receptor is to tell that cell it's time to divide, we need more cells. And a final example of an ultimate result of a pathway is something called apoptosis. Spelled apoptosis, and some people say apoptosis. I think it's supposed to be apoptosis. I think the second P is silent. But apoptosis is programmed cell death. And yeah, that's what the key terms read. Just those three words, apoptosis, programmed cell death. Obviously, there's a lot of complexity to this, but this is a cell going through a very controlled demolition. It kind of looks like a Death Star style explosion in some of these figures, but this is a controlled demolition as the cell very deliberately rips itself apart. Why would a cell do this? Well, if a cell is on its way to becoming cancerous or is infected with a virus, you want it to destroy itself to prevent it from spreading this infection or spreading this cancer or, causing harm to the overall organism. So a lot of times this is the final result of a message being sent to the cell. You need to destroy yourself through this highly regulated process. Okay. So far, all of these examples that I've been using, whether it's talking about cancer or whether it's been talking about hormones or things like that, or the bloodstream, so far all of my examples for this whole thing have been talking about multicellular organisms, big things like animals that have different cells and communicating across the body and stuff like that. And yes, there is a lot of communication that this is from a eukaryote here. There's a lot of communication that has to do with multicellular organisms. We are very complicated. We have very complicated ways of communicating. But the last section in this chapter, I want to focus on smaller organisms, because these same principles, these same basic things that we've been talking about are also going on in single-celled organisms. So, a couple of examples of this. One of these occurs within yeasts. So, yeasts are single-celled fungi, and they don't have male and female yeasts. They have A strains and alpha strains. But in order to complete this part of their life cycle and mix up their genetics, these two need to find one another. And yeah, these are single cells. They don't have eyes. They don't have ears. How are you going to find a suitable partner? Well, you're going to do it with ligands. So the alpha strain sends out ligands that are basically just saying, hey, there's an alpha strain here, there's an alpha strain in the environment, and the A strain sends out ligands saying, hey, I'm an A strain, I'm looking for alpha strain partner, and if these cells receive the ligands from the appropriate partner, that's going to tell them to start moving closer to one another, and they're going to complete this mating cycle. Again, it's really weird, can't get into the details here, but yeah, they're going to complete this part of their life cycle, mix up their genetics and split. So yeah, the way that they find one another here to do this is just through ligands and receptors, everything we've talked about so far. So here's my summary. Single-celled yeasts secrete what is called a mating factor ligand in order to find other yeast cells to mate with. Simple as that. Let's look at a more complicated example, though. Let's look at bacteria. So some bacteria can talk with one another. The most well understood example of bacterial communication is something called quorum sensing. So quorum is a very science-y sounding word, but it's actually not a science word. It's from meetings and politics. I don't know what to call that branch of study. I'm going to read the definition of quorum from the dictionary, not the textbook, because this isn't a science thing, from just, Googling what quorum means. Quorum is the minimum number of members of an assembly or society that must be present at any of its meetings to make the proceedings of that meeting valid. It's a bit of a doozy, but if you've ever been in student government in high school or college or anything like that, you know that these meetings are going to start by taking a roll call. Why is it important to see who's there and who's not there? Well, if only 3 people showed up to the meeting, those 3 people shouldn't be able to steamroll everything and make massive decisions for the entire body. You need a bunch of people there in order to make valid decisions. That's what quorum means. So what could this possibly mean with bacteria? Why do bacteria need to call roll at their meetings to determine how many people showed up? Well, they actually do. So, bit of a backstory here. Some bacteria can form structures called biofilms. This biofilm is a protective structure that surrounds a colony of bacteria, and from their perspective, this is fantastic. This protects them from drugs, from antibiotics. This protects them from the host immune system. They're safe within this biofilm. From our perspective, this is horrible. In a medical setting, in a hospital, biofilms are really dangerous to have to deal with. Again, drugs, antibiotics have a difficult time penetrating this biofilm and killing these bacteria. Our immune system can't reach these bacteria. This is bad news for us. The only good thing about biofilms from our perspective is that they are not easy to form. One bacterial cell cannot just form a biofilm around itself. It needs a bunch of buddies all together, working together to form this protective structure. So one cell can't do it, a bunch of cells can. So maybe you could see why the quorum is coming in here. They do need to call roll because they need to determine, again, they don't have eyes, they don't have ears, they need to determine whether they've got friends around them or whether they're more or less alone. So the ligands that these bacteria are going to release are called autoinducers. And these autoinducers are, again, they're the raise your hand, present, I'm here. These autoinducers communicate cell density to other bacteria surrounding them. So to summarize what I said about biofilms, some bacteria can form biofilms, but this requires a lot of cells to do, high cell density to form. The key terms define biofilms as a matrix of proteins and sugars that protects colonies of pathogenic bacteria from the host immune system. If you're curious, Pseudomonas aeruginosa is one of the more famous biofilm or infamous biofilm formers in a medical context. But yeah, some bacteria can do this, but they need a lot of friends to make it happen. It would waste energy to try to form this biofilm with few bacteria. Again, the key terms define this as a matrix of proteins and sugars. So this is formed essentially by ejecting proteins and sugars, just launching out proteins and sugars. You're throwing away these valuable energy molecules. It's worth it if you're actually going to form a biofilm, but it's an absolute waste of energy to try to do this if you're not actually going to form biofilm. You're just throwing away energy molecules. So in quorum sensing, we have 2 possibilities here. Either the ligand density can be low or the ligand density can be high. In the case of low ligand density, the ligand is just going to diffuse out. So here's the sort of side by side. We got the bacteria planktonic. If they're floating around in liquid or whatever, if they don't have a lot of friends, they are releasing this autoinducer, but it's just diffusing into the liquid around them. They know they don't have a lot of friends around. They're not going to try to make biofilms because that would be a waste. Low ligand density, ligand diffuses, no response. However, in a high ligand density, they are all sending out autoinducers, and they're all receiving autoinducer signals. Because of that, they know that they have a lot of partners around. They're going to begin the process, the energetically costly process of forming biofilm. So at a high ligand density, their signaling pathways are going to be activated, and there's going to be formation of biofilm. And again, all of this was to protect them from us and to prevent them from wasting energy. Again, for bacteria, wasting energy is one of the worst things they can do. So they're using these ligands and receptors to determine whether they have quorum or not, whether they should go for the biofilm or don't go for the biofilm. And so, yeah, understanding things like this is very important. There are a lot of drugs being developed now to try to combat this signaling pathway. If we could shut down their ability to form biofilms, their ability to talk to one another, to send and receive these ligands, that effectively shuts off their ability to form these biofilms if they can never establish a quorum. So again, this type of structure and things like Pseudomonas aeruginosa, very impactful in a medical setting, very important to understand more about biofilms and quorum sensing and how these things all talk to one another. Okay, so that brings us to the end of lecture 2.5, and yes, this is 5 lectures for each exam. So yeah, this is the end of lecture 2.5, and this, the end of the cell communication chapter. This is our cutoff for the end of the material for exam number 2.